Have you ever wondered what roles mRNA and tRNA play in the construction of our genes, or how protein synthesis is involved within this? Well, let's begin with what is protein synthesis? Protein synthesis involves DNA and RNA, and it takes two steps, transcription and translation. Transcription takes the information encoded in DNA and encodes it into the mRNA, while translation uses mRNA to work with the ribosome and tRNA to synthesize proteins. Transcription begins with the unwinding of the double helix of the DNA molecule so that this portion of the DNA can be transcribed. Once the DNA is unwound, the enzyme known as RNA polymerase helps to line up nucleotides with others so that they form complementary pairs. These complementary pairs form the strands of mRNA, and since mRNA is a single-stranded molecule, one of the strands of DNA is needed to be replicated in the new RNA. In translation, the mRNA is sent to the cytoplasm where it bonds with ribosomes. These ribosomes have binding sites, but the most important ones are the two tRNA sites labeled the A site and P site. Once the mRNA is in place, tRNA molecules that are associated with specific amino acids bind to the ribosome. They bind to the ribosome in a sequence defined by the mRNA code. When the mRNA chain binds to the ribosome, the first codon, which is always methionine, fills the P site, and the second codon fills the A site. The tRNA molecule, whose anticodon is complementary to the mRNA, forms a base pair with the mRNA in the A site. A peptide bond is formed between the amino acid attached to the tRNA in the A site and the methionine in the P site. The ribosome can then slide down the mRNA so that the tRNA in the A site moves over to the P site, and a new codon fills the A site.